Hi, my name is Mariah, and in this video, I'll be presenting a study of Chou and collaborators in 2020, the genetic diversity and population genetic structure of red pandas, Ailurus fulgens, in zoos in China. So to start off, we need to know where red pandas are found in order to conserve them. Red pandas are found in 5 different countries: India, Bhutan, Myanmar, Nepal, and China. The IUCN has considered this species as endangered due to habitat loss and fragmentation. For us, as biologists or ecologists, we have different conservation strategies. So in situ strategy is when we consider the whole ecosystem where the species is found and we conserve it. And the ex situ strategy is when the species is out of its natural habitat, and we mainly use it for purposes of investigation, conservation, but also education. This could be done in zoos or wildlife rescue centers. In previous studies, Liang in 2007 found out that wildlife population had a higher genetic diversity. And 13 years later, Chou and collaborators, they made this research question. Could a strategy of management and breeding programs in China's zoos maintain a versatile genetic structure for red pandas? So to do so, he took 166 captive individuals of red pandas from 11 zoos and examined 19 microsatellites loci for genetic diversity analysis. So the methods used were sampling, DNA extraction, PCR and genotyping, and statistical methods. For sampling, they took the red panda manual care from the Association of Zoos and Aquariums. From these individuals, they took 100 blood samples, 13 skin samples taken from the outer layer of the ear, 3 hair samples from the bottom layer of the tail. So in results, we have the comparison between genetic diversity of the captive and wildlife population. The mean number of alleles is a critical indicator of genetic variation. We can see that the Wyoming has a 7.1 number of alleles, the same as the Fuzhou Zoo that has 7.1. And we can see that the heterozygosity observed and expected vary in few decimals, not really much. And for the wildlife population has a similar genetic diversity as the captive population. So we see 9.2 and 9.3, being slightly diverse, the captive population. On the other hand, we have the inbreeding coefficient that for 6 zoos out of the 11 are showing that there could be a heterozygote deficiency. And the Hardy-Weinberg equilibrium could be showing that there is not a random mating of this population in these 2 zoos, Fuzhou and Wuxi Zoo. There is also the genetic differentiation coefficient that shows that there is a moderate differentiation between the zoos. But we have that the Mianning Zoo and Dazhou Zoo are very different in this case. In other results, we have the Bayesian genetic clustering for 11 captive populations. So here we have the zoos, and we can see that the Shanghai Zoo has a very very pronounced genetic structure, which is shown in these blue bars. So we could infer that maybe the Shanghai Zoo led to other mates in other zoos, knowing to transferring partners to reproduce in other zoos. So red pandas have a short life expectancy, a high mortality rate, making this more difficult for us to conserve in ex situ conservation. And we found out that there was a moderate genetic differentiation in the population, and also a low level of inbreeding in the total population. So finally, the study presented showed that zoos and wildlife population, they actually have a similar genetic diversity, and for captive population, well, it's a bit higher. And this means that red pandas could be progressing and could be reproducing, even though there's more investigation need to be done on the genetically part. And this is it.